Welcome to a new video tutorial series from Biology Boost Up. In this video series, we going to study about a new topic that is from biotechnology, and in this first video, we going to study about some terminology regarding biotechnology. So now let's start. First, the restriction enzyme. A restriction enzyme or restriction endonuclease is an enzyme that cleaves DNA into fragments at or near specific recognition sites within molecules known as recognition sites. Restriction enzymes are one class of the broad endonuclease group of enzyme. Restriction enzymes are commonly classified into 5 types, which differ in their structure and whether they cut their DNA substrate at their recognition site, or if the recognition and cleavage sites are separate from one to another. To cut DNA, all restriction enzymes make 2 insertions, once through each sugar phosphate backbone of the DNA double helix. Next one is restriction site. The restriction site or restriction recognition site are located on a DNA molecule containing specific about 4 to 8 base pair in length sequences of nucleotides which are recognized by restriction enzyme. Those are generally palindromic sequences because restriction enzymes usually bind as homodimers, and a particular restriction enzyme may cut the sequences between 2 nucleotides within its recognition site or somewhere nearby. Next, the palindromic sequence. A palindromic sequence is a nucleic acid sequence in a double-stranded DNA or RNA molecule wherein reading in a certain direction, for example 5 to 3 prime on one strand, match the sequence reading in the same direction, for example 5 to 3 prime on the complementary strand. This definition of palindromes thus depends on complementary strand being palindromic of each other. Now next, BT crops. Crops that are generally engineered to carry a gene from the soil bacterium, that is Bacillus thuringiensis. The bacterium produce proteins that are toxic to some pests but non-toxic to humans and other mammals. The crops containing the BT gene are able to produce this toxin, thereby providing protection for the plant. But corn and BT cotton are example of commercially available BT crops. Next, what is chromosome? The self-replicating genetic structure of cell containing genes which determines inheritance of traits. Chemically, each chromosome is composed of protein and a long molecule of DNA. Next, the clone, a genetic replica of an organism created without sexual reproduction. Next, cross-pollination, fertilization of a plant with pollen from another plant. Pollen may be transferred by wind, insect, and other organisms or humans. DNA, the chemical substances from which genes are made. DNA is a long double-stranded helical molecule made up of nucleotides, which are themselves composed of sugar, phosphate, and derivatives of 4 bases, that is adenine, guanine, cytosine, and thymine. The sequence order of the 4 bases in the DNA strand determine the genetic information content. Next point, the enzyme-linked immunosorbent assay or ELISA, a technique using antibodies for detecting specific proteins used to test for the presence of a particular genetically engineered organisms. Now next, the gene mapping, determining the relative physical location of a genes on a chromosome, useful for plants and animal breeding. Next, gene sequencing or DNA sequencing, that is determine the exact sequence of nucleotide base in a strand of DNA to better understand the behaviors of a gene. Next, genetic engineering, manipulations of an organism's genes by introducing or eliminating or rearranging specific genes using the method of modern molecular biology, particularly those techniques referred to as recombinant DNA technique. Now next, genetically engineered organism or GEO, an organism which is produced through genetic engineering. Now next, genetic modification, the production of heritable improvements in plants or animals for specific uses via either genetic engineering or other more traditional methods. Some countries other than the United States use this term of refer specifically to genetic engineering. Now next, the genetically modified organism, an organism produced through genetic modification. Next, genomics, the mapping and sequencing of genetic material in the DNA of a particular organism, as well as the uses of that information to better understand what genes do, how they are controlled, how they work together, and what their physical location are on the chromosome. Next, the genomic library, a collection of biomolecules made from DNA fragments of a genome that represents the genetic information of an organism that can be propagated and systematically screened for the particular properties. The DNA may be derived from the genomic DNA of an organism or from DNA copies made for messenger RNA molecule. A computer-based collection of genetic information from those biomolecules can be a virtual genomic library. Next, the genotype, the genetic identity of an individual. Genotype often is evident by outward characteristics but may also be reflected in more subtle biochemical ways not visually evident. Next, the PCR, a technique used to create a large number of copies of a target DNA sequences of interset. One used for PCR to detection of DNA sequence that indicate the presence of a particular genetically engineered organism. Next, the promoter, a region of DNA that regulates the level of functions of other genes. Next, the protein, a molecule composed of one or more chain of amino acid in a specific order. Proteins are required for the structure, functions and regulations of the body's cell, tissues and organs, and each protein has a unique function. Next, the recombinant DNA or rDNA, a small molecule of DNA formed by joining different DNA segments using recombinant DNA technology. Next, the recombination DNA technology. It is a procedure used to join together DNA segments in a cell-free system. For example, in a test tube outside the living cells or organism. Under appropriate conditions, a recombinant DNA molecule can be introduced into a cell and copy itself or replicate either as an independent entry or as an integral part of cellular chromosome. Next, selectable marker, a gene often encoding resistance to an antibiotic or an herbicide introduced into a group of cells to allow identification of those cells that containing the genes of interset from the cells that do not. Selectable markers are used in genetic engineering to facilitate identification of cells that have incorporated another desirable trait that is not easy to identify in individual cells. Next, selective breeding, making deliberate cross or mating of organisms so that offspring will have particular desired characteristics derived from one or both of the parents. Now next, the traditional breeding, modifications of plants and animals through selective breeding. Practices used in traditional plant breeding may include aspects of biotechnology such as tissue culture and mutational breeding. Now next, transgenes, a gene from one organism inserted into another organism by recombinant DNA technique. Next, the vector. In molecular cloning, a vector is a DNA molecule used as a vessel to artificially carry foreign genetic material into another cell, where it can be replicated or expressed. A vector containing foreign DNA is termed recombinant DNA. The four major types of vectors are plasmid vector, viral vectors, cosmid vector, and artificial chromosome. Of those, the most commonly used vectors are plasmid vector. Common to all engineered vector are an origin of replication, a multi-cloning site, and a selectable marker.